Hi everyone, my name is Precious Dialect and this is my presentation on Escherichia coli gastroenteritis. So just a few learning goals for us today as we learn about E. coli. We're going to talk about its discovery, a little bit of history of how it was um, found out. We're going to learn about the taxonomy of E. coli. We're going to differentiate the six pathotypes of E. coli and then explain the pathogenesis and epidemiology of some of those strains. And then most importantly, we're going to talk about ways to treat and prevent E. coli infections. So first things first, we're going to talk about the physician, the guy that, you know, helped us out the most with this type of bacteria, and this is Theodore Esserich. Hopefully you can tell from the name that that's where we got the name E. coli from, the Escherichia coli. But anyway, he was a German physician born in 1857. He studied at the University of Würzburg with a strong interest in pediatrics, and he focused a lot on the intestinal bacteria of infants. So, as he was doing his studies and as he was exploring his big interest on pediatrics, he actually had to go to different places around Europe in order to do that because during that time, Germany did not have a pediatrics unit. So he went to places like Vienna where he can really learn about what the pediatrics section means, what are some things that he can bring back to his own hospital and stuff like that. So that was kind of an interesting part. So during his work, he was able to isolate 19 different bacteria, and that includes bacteria coli commune, which is again now known as E. coli, and bacteria lactis aerogenes, which is Klebsiella pneumoniae. So he did that with all of the tools and tricks that he learned as a scientist, as a physician, to help distinguish and help identify what those bacteria are, what their characteristics are, and stuff like that. So once he was back to Germany, he started, just getting the pediatrics unit going. And then eventually, as he continues his work, he was nominated the chair of pediatrics at the University of Vienna and was the director of the St. Anna Children's Hospital in Vienna as well. So that's kind of an interesting tidbit because, again, Germany did not have a pediatrics unit, and because of his love and his interest in small children and infants and their welfare, he was able to start that for the country pretty much. So here's a little bit about the taxonomy of E. coli. E. coli is something that we commonly see in a lot of textbooks. When we talk about bacteria and structure, usually we use the E. coli picture to teach that to students because, again, of how well detailed and how well researched this bacteria was. So here is our breakdown. We have a domain bacteria, right? Phylum Proteobacteria, class Gamma Proteobacteria, order is Enterobacterialis, family is Enterobacteriaceae, genus is Escherichia, and the species is Escherichia coli. Again, just kind of a reminder, since it is in the domain bacteria, that means that they do not have membrane-bound organelles as we do as eukaryotes. Let's take a closer look at the structure of this bacteria. And again, you probably have seen that diagram, even, if all you've taken is basic biology, you've seen that diagram before. And so we're going to go through it in a little bit more detail so that we can take a look at, the special characteristics of this bacteria. So I'm going to give you guys all this info and then I'm going to kind of walk through them with you. So the shape of the bacteria is bacillus or bacilli for plural. That means rod shape, and that's kind of that pill looking shape right here that we see. It is a gram-negative cell wall. So again, just as a reminder, prokaryotes do have a cell wall. And we can see that right here in this label, cell wall, it is right below the capsule or the slime layer. An interesting characteristics for bacteria, they do have three layers. So we've talked about the cell wall. They do have a cell membrane as we do. Their cell wall is primarily made of peptidoglycan, and a bacteria cell wall can either be positive or negative. And in this case, E. coli's cell wall is gram negative. And then it also has an outer layer of capsule or slime, which is just extra protection for the bacteria cell itself. This particular bacteria has a flagellum, that is this long structure right here. That is what they use for movement. And it's not shown in this picture, but there are some E. coli strains where they have more than one flagella, right? That means that, there's more than one of these kind of floating around in that space. Just diving into a little bit of the internal structure, again, they do not have membrane bound nuclei. So this is their chromosome right here, kind of just floating around. They have ribosomes to help them synthesize their proteins. And then the very, very important plasmid. So plasmid is my way of saying like special abilities for for the bacteria. This is where usually the bacteria gets their antibiotic resistant properties. Sometimes they share that information between bacteria, or they just pick that up from a dead bacteria. Last important part of this cell structure is the fimbria for the attachments to host. It's not properly shown in this diagram. So when we get to talking about the attachment to the host, I will point that structure out to you as well. So one of the cool things about E. coli is that there's actually six different pathotypes. So pathotypes just means that there's six different ways that this same strain of bacteria can be infected, or sorry, that it can infect the host. So what that means is that there's different modes of infection. So usually E. coli is a commensal bacteria, commensal meaning that it doesn't hurt or help a particular organism. But in the case of the pathogenic E. coli, that's the one that can get you sick. If you've ever had food poisoning, that could be E. coli related. So let's take a look at the differences between these six pathotypes. So here's a diagram, a summary diagram of what they all do and how they all infect. I'm going to talk about them one by one so that the diagram is not too intimidating for you. But this is kind of all of them all together. So when you see these abbreviations right here, those are the E. coli pathotypes right there. So when it comes to pathogenesis, they have a main steps, right? Five main steps. So the adhesion, which again is attaching to the host cell. They need to colonize the mucosal site. They need to evade the host defenses in order to multiply, and then from there, um, give the host some damage. So as we go through each of the pathotypes, it's the same mode, only again, the style of how they infect are different. So I mentioned earlier about the fimbriae. So here, these blue structures right here are the fimbriae, and they use that as a way to latch onto their host. So here's the first one, enteropathogenic E. coli. They attach to intestinal walls, they eliminate microvilli, they stimulate the host cell actin, and then secrete effector protein. So if we're taking a look at this diagram right here, right, so these 10 projections right here are the microvilli of your intestinal walls. As you can see in this section right here, the bacteria has already taken off those microvilli, right? And you can see here too that they are starting to do the multiplication part. One of the cool, interesting things again is that they do stimulate the host cell, right? So this is the host cell's actin to form that pedestal, and they bring the E. coli up And from there, right, the symptoms of that would be diarrhea. You'll notice that most of these, the symptoms will be diarrhea. It's just going to determine, how bad or how watery the diarrhea is. For this next one, enterotoxigenic E. coli, same thing. They're going to attach to the microvilli. The main difference here is that they do not do the pedestal thing. They just attach to the microvilli and then start up a signal transduction that creates the enterotoxins and causes watery diarrhea. This next one, the enteroaggregative E. coli, again, more similar to the last one we just talked about. But the cool thing about this particular one is that they have a biofilm formation. So that's where we get the word enteroaggregative in there, is because they aggregate and they create that biofilm there. So they do create that stack brick configuration, just like you see here. They're not invasive, meaning that they kind of just stay up there, but they also do cause watery diarrhea. Then we have the entero-invasive E. coli. Now, this one is the only type of E. coli strain that does get internalized, right? If you notice from the previous slides, all of them kind of just stay up, but this particular one does get taken into the cell, right? Which then causes a lot more serious symptoms, right? As you can see there, inflammation, fever, and dysentery. And as you can see in this diagram, they can also migrate to different cells. So it's a lot harder to treat because they are within our cells themselves. The next one is the diffusely adherent E. coli. So as you can see from the picture, this one does not get internalized. However, they do kind of take over the cell's machinery to have that finger-like projection kind of hold on to them. So this is their special way of attaching to the host cell, right? Kind of just giving it a nice big hold right there. But again, also not internalized, not like the previous one. And then we have our enterohemorrhagic E. coli, which is one of the strains that we will talk about in just a little bit. This one is more harmful, and though the mechanism for their infection looks a little bit simpler looking at that diagram, the thing that really makes them dangerous is the sugar-like toxin that they are producing. That sugar-like toxin is harmful to the body, and that's where it gets, the very serious bloody diarrhea and stuff like that, where it could actually be life-threatening. So that's particularly because of the sugar-like toxin. So let's talk about this one in more depth. So again, this is the STAC, the sugar toxin E. coli. It's also known as O157H7. And again, this is the ones that are related to food poisonings when there's like a food outbreak, like when there was a point in time where there was like a lettuce E. coli outbreak and stuff like that. So this would be the bacteria that is responsible for that. So you can see the infected groups. It could be anybody, but the ones that have the most adverse effects are young children and elderly. The symptoms are right there. Again, same thing, diarrhea, stomach cramps, vomiting, and it can spread through contaminated food or unclean food. So if you have, let's go with the lettuce example. If you eat lettuce and it had E. coli in it, then you can get that poisoning. It could also go indirectly. So for example, if let's say a cow eats grass that has E. coli and then you go ahead and ingest the meat of that cow, then you can also get the infection that way as well. Treatment and prevention, hydration is key, right? You want to drink water. Diarrhea is great loss of liquids from your body, so you want to stay hydrated as much as possible. For this particular type of E. coli, antibiotics has not shown evidence that it works to treat it. And same thing with antidiarrheal agents. The reason for this is because you are increasing the risk of hemolytic uremic syndrome, which is kind of a serious thing for a staph infection. So usually you kind of just have to write it out if the symptoms are not super big, but you do not want to treat it with any antibiotics or antidiarrheal for this particular strain, right? Prevention tips right there for you. Honestly, a little bit of a common sense there in terms of prevention. Washing hands definitely important, and cooking your food thoroughly will also help kill off those excess bacteria that may have potentially gotten into your food. The enterotoxigenic E. coli is the common cause of travelers' diarrhea. So it's going to be the same kind of symptoms as the stick. And it's the same mode of spread where it's food, drinks, contaminated water. And the same treatment. So this one, though, you can treat with antibiotics. You are still gonna have those symptoms of diarrhea and dehydration and stuff like that, but the antibiotics can help shorten the duration of the infection. Um, One of the reasons again is because of the stuck one has the sugar like toxin, which is really what kind of brings up all of those symptoms, whereas the enterotoxigenic, not as bad, right? Not as bad. Same types of prevention for both. And really for any type of bacterial infection, that's kind of your best mode of prevention. Washing hands, cooking meats thoroughly, not eating raw meats or raw milk, right? And preventing cross contamination. And there you have it. Thank you guys for listening to my presentation. I hope you guys learned something new, and see you guys next time.